He is the Nobel Prize winner for medicine, known for his discoveries concerning nitric oxide, and Director Emeritus of Brown Foundation, Institute of Molecular Medicine for the Prevention of Human Diseases. I'm pleased to welcome Professor Dr. Farid Murad for his keynote address. Professor.
Well, thank you. I would like to welcome all of you to our third international meeting for drug discovery and therapy. From the program that I have reviewed, I expect that this meeting will be even better and more popular with information for all of you than the previous two meetings. It's looking like a very good meeting from the titles of the presentations and the list of speakers. I would like to also thank His Excellency, the Minister of Education and Science, for helping support this meeting and for all of you to be here. There are several, I'm going to keep my comments very brief. There are several approaches to discovering new novel drugs. Some years ago, what we would do is to take chemicals off the shelf or that we synthesized in the laboratory and put them into a biological system, an intact animal, cell cultures, some living system, and observe the physiologic response to see whether it looked interesting and beneficial, hopefully without some toxic effects in the system that we were studying. And if that was the case, then those compounds would be pursued for possible candidates for clinical development for future use. The process of drug discovery has changed dramatically in the last several decades. What we now do is discover molecular targets. And we discover these targets in a variety of ways with various technologies, including biochemistry, pharmacology, physiology, cell biology, other approaches, genetics, etc. And we define pathways that regulate some important biological process that we want to perturb and modify. And we take often those rate-limiting enzymes and proteins in that pathway as potential molecular targets, and then we screen them against various chemicals. And today, we can screen them against huge chemical libraries. Some of these libraries are hundreds of thousands of compounds. Awful lot of screening, but invariably, it will turn up with a number of hits that we call potential drug leads that have an effect on that molecular target of interest, either as activators or as inhibitors. We then take those leads and perturb the structure in various ways with medicinal chemistry or computer programs to see if we can't design a molecule that will have optimal pharmacokinetic properties, formulation properties, and be a compound that can be made at the least cost, because the lower the cost, presumably, the greater the profit margins as well as the distribution of that drug someday. That is the mechanism that most of us entertain with drug discovery these days. I've been doing that now for some years, my entire career, as a clinical pharmacologist. We've discovered the mechanism of action of lots of drugs. We've helped design lots of drugs along the way. And once we design an optimal drug, what I often do is pass them on to collaborators and colleagues who will pursue the clinical studies, the clinical trials. Although I've done some of that in the past, I tend to emphasize the basic research myself for the last few decades. Another approach, which has a lot of expectation and promise, is to design a chemical hit or lead a potential drug, based on the physical chemical properties of that molecular target. And we think of the X-ray structure when we do that. If we can predict the X-ray three-dimensional structure of a protein, identify its catalytic pocket or binding site of some ligand, then we can go to the computer and begin to design molecules that will plug into that site as potential activators or inhibitors. That's been a hope for a long, long time. I was fortunate when I was at Abbott Laboratories 20 years ago to have had a talented virologist and X-ray crystallographer working for me who managed to crystallize the HIV protease, look at the X-ray structure, identify the symmetry in the catalytic site, and screen some of our peptide libraries to find a new HIV protease inhibitor that ultimately ended up on the market within several years. That program was really accelerated quite rapidly. Within a year and a half from start to finish, we had it in the clinic. And another year and a half or so, we had it on the market. Never had anything been done that quickly before. Is that going to be the future? I think so, but it's a tough nut to crack because there's a lot of flexibility in proteins. You can't predict necessarily with amino acid structure from DNA how you're going to get a compound that modify that activity. But that's the desire. So things will change even more. And we think we've all learned is that the whole process of biology, drug discovery, biomarkers, whatever, changes rapidly and continuously. And just think of how the fields have changed dramatically in the past decade. It's been remarkable. You know, to display proteins on gels and identify them so quickly and display genes, that's going to alter our whole process of disease diagnosis and therapy. When I was at Abbott, we did a study, screened the literature, did some surveys, and discovered that about 17% of the total revenue of each of the big pharma companies was utilized for R&D, research and development, 17%. About half of what was being spent for marketing and sales. I thought to myself, as a technology company, how can we spend so little and have marketing spend so much if we had really good products? Well, today, 20 years later, those numbers have changed very dramatically. And today, the big pharma companies, on average, are only spending 11% on R&D. I saw that coming because in industry, I saw the emphasis to do more phase 4 and clinical things and less discovery. And I said, my goodness, someday the pipeline's going to disappear and dry up, and that's exactly what's happened. If you look at the big pharma companies, they're acquiring the small biotechs, they're merging with other big pharmas because their big blockbuster drugs are coming off patent and their pipelines are empty because they haven't invested sufficiently in research. And that's got to change if we're going to be competitive in the future and find important drugs. Now, another prediction that I can share with you is that in the future, therapy is going to become very personalized. Most of the diseases that we're interested in, hypertension, heart failure, dementia, diabetes, cancer, are multifactorial. There are multiple causes. And if you take a population of patients with these syndromes, any one of them, their causes to get to these symptoms is going to be quite different. So we can no longer think about using a single compound to treat all these patients in such a category. There's not going to be a common drug for hypertension or for diabetes or whatever you're going to treat, because all of these patients will be different and heterogeneous. What does that mean in terms of sales? There will no longer be multi-billion-dollar products on the market some decades from now. There will be small markets with discrete therapy for patients with specific diagnoses who've had proteomic studies and genomic studies and biomarker studies to tell you what category they fall in, is how to treat them best, so that their drugs that you will give them will be most effective, handled pharmacokinetically the best way, and not metabolized and not toxic, and it's very specific. That's going to happen. There's going to be the day when a patient walks into the doctor's office and provides a swab of the mucosa from the mouth or a blood sample that goes into a machine, and an hour later they'll predict what drug this patient with hypertension or heart failure or diabetes or whatever should be treated with. Now, what is this going to mean to the pharmaceutical industry? They're going to get nervous about this because they're not going to have these huge, huge markets anymore. But on the other hand, their clinical studies should become much easier and much cheaper. I think that you can get answers about efficacy and toxicity, perhaps if you're lucky, with hundreds of patients or 1,000 patients and not thousands of patients, which they often do today. I've never understood why it's necessary to test 3,000, 5,000 patients. A few hundred is sufficient. The only time you need to do that is when the toxicity is so small that it takes a huge population before you see an effect. For example, if you have a drug that is toxic 1 out of 100 times in a patient, you're going to have to study hundreds of patients to see a significant number to convince yourself there might be a problem. So, and that's the toughest part, in my opinion, of drug development, is the toxicity, developing some ground rules as to what's likely to be toxic or not. The other parts, the drug discovery and all the mechanics of formulation and medicinal chemistry and designing clinical studies, pretty straightforward. In the time I was at Abbott, in 4 or 5 years, I just helped discover 24 new chemical entities to go in the clinic. That was exciting. It's doable, can be done. So times will change. I think there's a big need for all of these tools. I think you're going to see in this meeting the different approaches that can be taken to discover new chemical entities and leads to make drugs, whether it's computer modeling or physical chemical studies, molecular targets. All of this is feasible. And we welcome you, and I hope you enjoy the next few days. Thank you.